Okay, good afternoon. Let's get started. It's my pleasure today to introduce Bruce Ellis as part of our Bloomberg Distinguished Professor presentations. Dr. Ellis is currently the professor and the John and Doris Norton Endowed Chair in Fathers, Parenting, and Families at the University of Arizona. Dr. Ellis received his BS from Cal Poly and his PhD in psychology from University of Michigan. And he followed that with a postdoc at Vanderbilt in developmental psychopathology. So some of you may already be familiar with his work on biological sensitivity to context, particularly in children. Those of you that have ever read any articles that use the words dandelion and orchids to apply to children may recognize it. Dr. Ellis's work is at the interface of evolutionary biology and developmental science. He is the recipient of the George Miller Award, which is a psychology award from the American Psychological Association for best paper in conceptualization that spans disciplines. And it's a great honor and a career to receive this once. He received it 2 times. Dr. Ellis also, at one point in his career, although this is not in a CV, was the, I believe, southernmost personality psychologist in the world. Is that correct? Southernmost dwelling personality psychologist in the world when he worked in New Zealand. And also just showed me how to use PowerPoint notes to PowerPoint, what's it called? PowerPoint presenter. So at the screen, you can see your notes from a PowerPoint presentation. So you no longer need to bring paper. That's sort of mind-boggling. So without further ado, I'd like to introduce Dr. Ellis. Okay.
I was told there'd be severe consequences if I did not turn on my mic, so I just did that. So it's really a pleasure to be here. I've had a chance to meet and talk with several of you and to learn more about what's going on here in public health and in the School of Education, and to try to understand the linkages. I feel very honored to have been invited here to interview for this Bloomberg Distinguished Professorship. So thank you for having me. My talk today is going to be divided up into 4 parts, and that is I'm going to start off by talking about stress development and health, and I'm going to present the life history theory framework which I use to frame my work, so start off with a little bit of a theoretical background. I'm then going to talk about some of my empirical research on stress development and health, particularly in relation to the angle I bring to this, focusing on biological sensitivity to context, as Janet mentioned in her introduction. I am then going to shift to talking about some work that I'm doing on cognitive adaptations to harsh environments, looking at executive functions and learning in high-risk youth, which is actually closely related to the stress health development work as well. And then I'm going to conclude by saying, a few words about my vision or thoughts about the Bloomberg Professorship. So central to the study of human development are questions about variation in social behavior. And you can think about this, why do some people show high investment and commitment in many aspects of their lives? They're highly selective in choosing a mate. They provide extensive parental investment in their own offspring. They make long-term plans. They're good at delaying gratification. They have long-term committed relationships. They're more likely to be altruistic, cooperative, cautious risk takers, and to give and receive social support. Whereas others show little commitment investment in these various domains. They tend to have short-term relationships, provide little parental investment, plan only for the short term, focus on immediate rewards, and demonstrate less altruism, less cooperation, give and receive less social support, and engage in more impulsive, risky, and aggressive behavior. Of course, if I could actually answer this question, I could retire, but I'm not quite there yet, don't have retirement in the plans. But actually, these questions, these fundamental questions about variation and social behavior and adjustment are, of course, of great interest to social policymakers. Less widely recognized is that these types of questions, as well as related ones, such as relating to timing of puberty and family size, are also of central interest to evolutionary biologists as well. So there are multiple fields that you don't always think of that converge on some of these issues. Now, to actually answer these big questions about the development of the phenotype and variation and basic variation requires really a true integration. It requires bringing in developmental psychology, which is a field that really helps us to understand how rearing experiences affect development. It requires bringing in neurodevelopment, neurobiology to understand mechanism, to understand brain structure and function, neuroendocrine systems. And it also requires bringing in evolutionary biology to understand why rearing experiences actually operate the way that they do. And so in this talk, the goal of my talk is to try to tie these three areas together to try to move towards something of an answer to those questions. And particularly, I'm going to try to tie these areas together to understand adaptations to stress, and particularly how adaptations to stress guide variation in the developing phenotype. Now, first off, in understanding developmental adaptations to stress, we can kind of think about the traditional public health models. And these are models that you would all be familiar with. And these traditional models have many names. They're sometimes called cumulative stress, or toxic stress, or allostatic load. But they have in common that all the bad stuff goes together. And so from these models, high stress environments lead to exposure to high stress environment, to abuse, to neglect, to poverty, lead to disruptions of brain structure and function. These disruptions ultimately dysregulate physiological mediators and lead to impairments or disturbances in development. And so this perspective focuses on the destructive effects of dysfunctional families and harsh ecological conditions on mental and physical health. And from this perspective, from this kind of cumulative stress perspective, dysfunctional behavior in childhood and adolescence is seen as a natural consequence of exposure to harsh, unpredictable, and uncontrollable conditions. Now, in contrast, consider an evolutionary perspective on developmental adaptation to stress. Now, from this perspective, risky behaviors and cognitions reflect adaptations to risky environments, not deviations from optimal development. And in this context, thinking about this, stressful environments have always been part of the human experience throughout our history. In fact, when you look at hunter-gatherer societies, which are the best model for human demographics before the agricultural revolution, what you see is about half the children that are born die before they grow up. Stress has always been with us. Stress, real stress, serious stress has always been part of the human condition. And so when we encounter stressful environments, this should not so much disrupt or impair development as direct or regulate it towards strategies that are adaptive under stressful conditions, even if those strategies have trade-offs that involve costs to mental and physical health. And what I'm going to argue today is that we need to understand these functional adaptations to stress to actually understand dysfunction, to understand what those trade-offs and consequences are. And in understanding adaptations to stress, a major framework for explaining this is life history theory. And life history theory is a theory from evolutionary biology that is used to explain how individuals adapt their physiology and behavior to different ecological contexts. And in life history theory, the key concept is the concept of trade-offs. And this is that basically you cannot do it all. The amount of time and resources that individuals can devote to major life domains is limited. So organisms have several things they need to achieve. They need to achieve growth, just development of size, strength, coordination, skills, knowledge, all these investments such as we make in educating ourselves. We need to invest in survival, all the things we do to allay mortality, DNA repair, immune system, and we also invest in reproduction, finding a mate, producing offspring. And we can't invest the maximum in all these things at once. We have to prioritize resource expenditures. So we're selected to make trade-offs that we're selected to make trade-offs that prioritize how we expend resources in these different domains. Now, these trade-offs are instantiated in key developmental decisions over the life course. One key developmental decision is earlier versus later reproductive development. So, for example, resources that are devoted to fast growth and early puberty cannot be spent on longer-term somatic investments. So the benefits of early puberty are actually traded off against costs to mental and physical health as the organism matures. Likewise, there's trade-offs between resources devoted to survival versus reproduction. So resources spent on an inflammatory host response to fight infection cannot be spent on reproduction. So the benefits of this inflammatory host response are traded off against the costs of lower fertility. Now, in this model, each one of these trade-offs constitutes a decision node in allocation of resources. And each decision node influences the next, opening up some options and foreclosing others in an unending chain over the life course. And this unending chain of decisions defines alternative developmental pathways, or what are called life history strategies, that are matched to ecological context. Now, in thinking about life history strategies, it's actually useful to think about two kind of broad strategies. And these two broad strategies can be represented here in terms of these two pathways. And over here, this is, you can think about this fast strategy, this being a high stress pathway over here, a slow strategy, this being more of a developmental pathway that you see in a stable, supportive environment. And each one of these represent physical and behavioral and psychological adaptations to stress. And this is ultimately what I do. I study adaptations to stress. And over here, this is adaptations to a high stress environment. This is an ecological and family context characterized by poverty, characterized by parental instability and change, characterized by high stress and danger. And individuals, parents in this context, this is the kind of high stress context that often exceed the regulatory capacities of parents to deal with them. And parents in these contexts have less resources left to devote to invest in their own offspring. So it often undermines quality of parenting. Parents under these conditions are more likely to engage in harsh, rejecting, insensitive, inconsistent parenting. Well, this parenting itself, the types and the levels and types of parenting that children are receiving, this is actually a conduit to the children. It's communicating to the children the type of ecology or environment that they're growing up in. It's associated with more, from the child's perspective, more insecure attachments, more development of more risky and aggressive behavior, but also cognitively a focus on things like more immediate gratification, more shifting attention rather than sustained attention between different things in the environment. In turn, according to this theory, this high stress pathway is associated, when you're growing up in an environment where the future is uncertain, the future is unknown, you tend to develop in a manner that speeds up development, that tend to accelerates puberty, accelerates onset of sexual activity, accelerates reproduction. So this is a high stress, fast developmental pathway. It's associated with less somatic effort. That is, you're putting less effort into DNA repair, you're putting less effort into certain forms of immune function, and as a result, it's associated with more health deterioration over time. And over here, this is just the other side of the coin. This is a stable, well-resourced environment where that supports higher levels of parental investment by the parents, and among the offspring, adaptations to a stable environment such as inhibitory control, secure attachment, orientation towards long-term goals, long-term investments, later development, more investment, more somatic effort, more investment in health. So, in thinking about this, in this diagram, there's no good or bad. There's no value judgment in terms of what's good parenting or what's bad parenting. Okay, from this, different forms of parenting are preparing children to grow up in different types of environments. And so from this perspective, a child who lives in a risky, unpredictable environment, who's developing insecure attachments and becoming mistrustful of others and engaging in more risky and aggressive behavior, is no less functional than a child who's growing up in a safe, stable environment and developing the opposite propensities. These are adaptations to different environments with different types of trade-offs. Okay, there we go. Okay, so this basic life history model is consistent with a lot of evidence showing that more harsh or unstable environments predict higher levels of externalizing behavior and earlier puberty and sexual activity and more health risk, and that conversely, more stable and supportive environments are associated with more pro-social behavior in puberty and later sexual debut and better health. And much of my research over the years has tested various aspects and components of this model. And here's an example of just the kind of empirical research that I typically have done, and I do many studies like this. And it often involves longitudinal research, so it involves studying, children and families over time. There is an assessment of family and ecological condition, and this is often done in preschool. Early in life, the first 5 years of life, we're assessing things like socioeconomic status, marital conflict, parental supportiveness. A lot of my work focuses on pubertal development, so around 10 to 12 years of age, you're often assessing, around 5th grade or so, assessing puberty. And then in this research, we are both assessing various intervening mechanisms. In this particular study, it was assessing BMI, and then also assessing various outcomes on this end in terms of sequelae, such as externalizing behaviors or risky sexual behavior. So here, this is a model that was, in this particular analysis, we were interested in timing of pubertal development. And this is a study of girls, and it was asking the question, why are some girls, what is regulating levels of pubertal development in these daughters? And this is, so this is one of the key, timing of puberty is one of the key variables in the life history model. And what you see here is we have, mother's age of menarche as a kind of control for genetic influences. And mothers, who had later age of menarche, their daughters showed less sexual development in the 5th grade. These are standardized regression coefficients. This is in a structural equation model where everything is fit. And here you can see that there are actually, so this is one of 4 pathways leading to pubertal development. You can see that we also have a measure of socioeconomic status in preschool, and girls that were growing up in more socioeconomically favorable positions, families with higher occupational status and parental education, they were showing, as measured in preschool, these girls were showing less pubertal development in the 5th grade. You also have here a measure of parental supportiveness. This is a composite of various measures of family support and family stress, and you can see higher levels of parental supportiveness associated also with later pubertal development. So this is like that slow life history strategy pathway where more family support, closer parent-child relationships as measured before the onset of puberty are subsequently predicting less pubertal development in the 5th grade. There's also a pathway, you can see, of course BMI predicts pubertal development. So in the 3rd grade, girls that were heavier BMI showed more pubertal development in the 5th grade. And we had an indirect pathway going from marital conflict and depression. So this is like more of a measure of the family ecology to which the parents are fighting with each other and there's parental depression. That is predicting heavier BMI, higher BMI in the 3rd grade, and there was an indirect pathway from marital conflict and depression to pubertal development. So children in more conflictual families, younger, were tending to more likely to gain weight. A whole literature about stress and eating behaviors and that itself. So we have 3 different things converging here to 4 different pathways converging to explain pubertal timing. And so these are the kind, this is an example of the kind of studies that I do, looking at psychosocial factors in the family and how they play, the role they may play in regulating neurobiological systems, particularly the HPG axis, the hypothalamus pituitary gonadal axis and related outcomes. And so I've tried to examine this question over my career. But if you actually look at this, what you see is that we've succeeded in explaining about 1/4 of the variance in pubertal development, that any one of these effects is relatively small. There's significant effects of SES and parental supportiveness and et cetera, but these are not big effects. So this kind of raises the question, not only does psychosocial, do psychosocial factors regulate pubertal timing, and this is the same thing that happens when you look at these psychosocial factors are, risky sexual behavior, child aggression, you get reliable but small effects. And so it raised the question, not just, is there a role for environmental regulation, but for whom? For whom does this actually matter? And this brings us to the topic of biological sensitivity to context. Now, in this work, it's trying to explain what do we do with these small effect sizes? How do we actually interpret this and deal with this? And a possible explanation for these small effects is that children may be differentially susceptible to their family environments. That is, the small main effects may actually underestimate the impact of rearing environments in some children and overestimate it in others. And this kind of core idea led us to develop this idea of biological sensitivity to context. And biological sensitivity to context is a concept we published in 2005 with my longtime collaborator, Tom Boyce. And we defined it as an enhanced neurobiologically mediated susceptibility to both risk-promoting and development-enhancing environmental conditions. So essentially, that's a verbal, a lot of big words there, but it's a way of saying that it's an openness or susceptibility to both positive and negative factors in the environment. And we indexed, in our work, we index and measure biological sensitivity to context in terms of activation of all three of the main stress response systems. So we measure it in terms of sympathetic nervous system, parasympathetic nervous system, and HPA axis activation. And in this work, we argue that biological sensitivity to context operates as a central mechanism of plasticity. That is, that it regulates alternative patterns of human development. And specifically, what we mean by that is that individuals that we measure and define as being high in biological sensitivity to context, that these sensitive individuals should be more likely to experience developmental change in response to environmental conditions. Now, in developing this concept, to try to communicate it, we developed the analogy of the dandelion and the orchid. And the dandelion child is actually a Swedish expression, and it's an expression that Swedes use to describe a highly resilient child. So a dandelion can grow on the cracks of a sidewalk. It comes up in your grass, you mow it down with your mower, you think you've got it, but it pops right back up the next day. It's very tough. And we borrowed that term from the Swedes, and we then, to try to show the contrast, we invented the concept of, or put out the concept of an orchid child. And an orchid is a flower that, given the right conditions, and my grandmother who lives in Florida, my 99-year-old grandmother who lives in Florida, grows orchids in her living room. And she has just the right levels of humidity and heat and moisture and salinity. And under her care in Florida, her orchids bloom into flowers of unusual beauty and delicacy. But given the same flower in my living room in Arizona, it would just, wilt and dissolve. And so we use the orchid child, so we developed this concept of an orchid child to represent the concept of a child that, given the right nurturing and support, can develop into, highly socially competent, effective, successful child, but the very same qualities that make them susceptible to a good teacher or a good parenting also make them susceptible to adversity and stress, and they'll show the worst outcomes. And to actually communicate this idea of the orchid and the dandelion, these are the kind of data that we use. And so the orchid and the dandelion data we always present in the same format. There's always an x-axis, which is the environment. And so here, in this the environment is marital conflict, from low conflict to high conflict, and this is in 5-year-olds and their families. And there's always a y-axis, which is some child behavioral outcome. In this case, it is externalizing behavior. And we're asking, does higher levels of marital conflict produce more externalizing in 5-year-olds? But then there's also the for whom. And the for whom is the moderating factor, and that is our measure of biological sensitivity to context. And in this case, the 5-year-olds, all come into the lab and they do a standardized laboratory protocol. They get cognitive stressors and social stressors, and we're getting baseline measures and reactivity measures of the stress systems. And this is showing parasympathetic reactivity, so we're measuring, so this is a measure of vagal withdrawal to the extent to which children, when they experience our stressors, are releasing that parasympathetic brake and showing activation of the system measured by RSA. These children right here, this is modeled at 1 standard deviation below the mean. These are low parasympathetic reactivity. These are the kids that we call the dandelions. Okay, and for them, you can see that high marital conflict, low marital conflict, it doesn't matter. Externalizing looks just the same. It's like water, it's kind of like water off a duck's back. These kids are resilient against bad conditions, they're resilient against good conditions, they just kind of are what they are. Now these right here are kids that are what we would call the orchid children. And these are ones that are modeled in this regression interaction at 1 standard deviation above the mean in parasympathetic reactivity. And you can see that for these kids, these are the ones who are really responsive to their environments. High marital conflict, they show the most externalizing. Low marital conflict, they show the least externalizing. They show these outcomes for better and for worse. Now here is the same, again, these graphs, we always have the same format to try to minimize the confusion level, but the x-axis, this is family adversity. This is a composite measure of kind of overall multiple measures of family adversity. This is a positive outcome here on the y-axis, which is school engagement at age 5. And I guess, and does family adversity predict school engagement? Well, here, the dotted line, these are our dandelions. And you can see that low family adversity, high family adversity, this line is pretty flat. It's certainly not a significant slope. They show about the same level of engagement. But for these kids, for the orchid children up here, who are modeled, again, this is parasympathetic reactivity at a standard deviation above the mean, you can see that under low family adversity, they show the most school engagement. Under high family adversity, they show the least school engagement. So this is, again, so in our theory, we're using activation of the stress response systems, the extent to which these things are changing from baseline, as our indicators of biological sensitivity to context. And we got excited about this concept of developing this idea because for a long time it was thought of, stress reactivity, that's a vulnerability factor. But what these kind of data show is that the very characteristics in children that we thought of as their greatest vulnerabilities can also be their greatest strengths, given the right environmental context. And that was, more or less the story or lesson of biological sensitivity to context. Okay, so coming back to this slide, we thought, well, let's take a look at these data and let's reanalyze them, thinking about, let's see if we can do a better job bringing in stress reactivity, bringing in biological sensitivity to context. So we took this relationship right here, this 0.22, beta weight, this negative 0.2 beta weight, suggesting that there was a main effect controlling for all these other factors. So there was a main effect where higher parental supportiveness was associated with, in kindergarten, in preschool, was associated with less pubertal development in the 5th grade. Well, among these children, when they were in 1st grade, we brought them in and did the same laboratory protocol where they came in, they did a set of standardized laboratory tasks. I'd be happy to talk to anybody at length about these, but I don't have time to go into them now. And we were, assessing, the same set of stress reactivity measures. And then we reanalyzed these data. We reanalyzed that particular relationship using a piecewise growth curve model. And what you have here, if you look at these data, these solid lines, in this case, we're looking at the interaction between parental supportiveness, high and low parental support, in this case modeled at a standard deviation above and below the mean, and sympathetic nervous system reactivity. This here was a PEP measure this time, looking at shortening of the PEP intervals in response to our stressors. And these are also modeled at standard deviation above and below the mean. And these solid lines show our dandelions. These solid lines show the kids who are a standard deviation, who are modeled at a standard deviation below the mean in biological sensitivity to context, low sympathetic nervous system. And you can see the red shows high levels of family support. The blue shows, oh, by the way, on the x-axis is pubertal development. And now we've had these kids over time. The model is centered at age 12 and a half. So this is showing pubertal development from age 9 and a half to age 15 and a half. And one golden rule about studying kids from 9 and a half to 15 and a half, you can say a universal puberty happens. So you can see that everybody went from low levels, this is Tanner stage over here, from their Tanner stage from being, all these groups were down here around 1 or 2, they all went up to around 5 by the end of the study. Okay, but for the dandelions, you just look at this, it's just smooth monotonic development. Each time they're assessed, they show a little bit more about the same rate. High family support, low family support, it doesn't matter, they look exactly the same. Now these dotted lines are the kids, are orchid children. And these are the kids that were modeled at a standard deviation above the mean in sympathetic nervous system reactivity. And the red shows high family supportiveness. And you can see that for these kids, the kids that were sensitive to their context and receiving highly supportive family environments, they showed hardly any change in pubertal development over this time period from 9 and a half to 12 and a half. And then they caught up in the second part of this analysis. Whereas the kids that were high and sensitive to context and received low family support, were in rather harsh conflictual family environments, they had the most accelerated puberty and then slowed down. So you see this large separation. So high versus low parental supportiveness only distinguished pubertal tempo, the speed of puberty, and pubertal timing in adolescents with high sympathetic nervous system reactivity. So again, it's coming back, the question is not just, do psychosocial factors regulate sexual development, but again, for whom do they regulate sexual development? Now, I just want to mention, and these are not my data, I want to mention that people that study biological sensitivity to context, it has a larger name, and the field is called differential susceptibility. And most people that study differential susceptibility, study actual genetic markers of it. So they're looking for susceptibility alleles that operate in the same way as our stress reactivity factors. So here is someone who is studying, just like I did, quality of family environments and age at menarche. And they're asking, do children growing up in higher quality this is work by Manick and colleagues, growing up in higher quality family environments experience later age of menarche? And they found, just like I found, that it depends, but in this case, they were studying a polymorphic variation of the estrogen receptor alpha. And they particularly found that individuals that were homozygous for the GG form of this allele were the ones that showed the strongest effect. So the reason I'm showing this is I want to put this in a bigger perspective, that biological sensitivity to context, what we call that, even though we've operationalized it and measured it in terms of the stress response systems, is actually instantiated in multiple genetic polymorphisms and multiple neurobiological mechanisms. Some people study it in terms of hippocampal volume. There's all kinds of different ways to study this. And behavioral phenotypes, all of which operate as susceptibility factors, create those same kind of crossover interactions. That seem to moderate the influence of environmental exposures on developmental and life outcomes. And if you want to see a really detailed review, I would point you to this paper. It's the 5 authors are kind of the 5 people that were most responsible for developing this field of differential susceptibility. The toughest thing I ever did was leading the 5 of them in a collaborative paper to spell out the field. That was a challenge, but hopefully a model for what you're looking for in a kind of Bloomberg professor who would try to pull people together and talk to each other and talk about concepts. So that paper, is a good point to get an overview of this literature. And this idea of biological sensitive context has far-reaching implications for understanding whether and how much child and adolescent development responds, for better and for worse, to the gamut of environmental conditions. And this is really thinking about intervention. This is a strong message that one size does not fit all. That any intervention that doesn't consider the child's susceptibility or openness to their environment is likely to waste an awful lot of money trying to do the same thing for everyone. And if we can get more sophisticated in understanding the extent to which different children will respond to different kinds of interventions, we can get a lot farther in terms of being effective in this domain. Now, coming back to this bigger picture, what I have been talking about is I've been looking at this pathway, or actually more looking at this pathway, because I was looking at high socioeconomic status, sensitive, supportive parenting, marital harmony, all of these things predicting in the model I showed you later onset of puberty. So this is regulating the reproductive axis. Now, I am also, in terms of going forward, in terms of where I'm going with this now, I wanted to show you that, to kind of show you the most basic findings to communicate the nature of this work. I'm very interested in understanding the implications for health trade-offs. Because, in these pathways, this high-risk pathway is very much about reduced somatic effort, less investment in health. This low-risk pathway is very much about more investments in health and health trade-offs. And so my current work in this area and the work that I have going forward, and that if I were to come here, in the stress health domain, I'd be very interested in seeking to understand the implications of biological sensitivity to context for understanding these health trade-offs in development over time going down the road. Okay. I want to shift now and go to the kind of the second major part, the other major part of my talk, which is talking about cognitive adaptations to harsh environments. And this is the part looking at executive functions in learning in high-risk youth. And this is a relatively newer research area for me, but an area that I'm very excited about and am now pursuing actively. So coming back to this model, when you look at this model, you see that it also involves not just quality of parenting and not just sexual development and health, but it also involves behavioral and psychological development, and it involves cognition. You can ask the question, what do children growing up in harsh, unpredictable environments, what do they actually learn? What are kids in these environments good at reasoning about? What are they good at remembering? What do they pay attention to? And so you can ask the question, does growing up in a stressful environment during childhood actually improve certain mental abilities? Well, the evidence thus far suggests that the answer to that is probably no. Because when we take these kids from high-risk environments and we test them, we test them under very standard testing conditions, standard sanitized laboratory conditions, they don't do very well on standard tests of intelligence or standard tests of working memory or standard tests of executive function. And this is consistent with a cumulative stress model, with a standard model that all the bad stuff goes together and that exposures to stress is impairing development and dysregulating kids. And that's half the story. But I'm going to argue today that that's not the whole story, and that rather than actually impairing mental functioning, childhood adversity could instead be shaping it in adaptive ways. And that people that are growing up under harsh environments may be good at solving problems that are ecologically relevant to them and their ecology. Now, executive function, just to give a very brief definition of executive function, refers to the management of cognitive processes that guide behavior towards various goals. And executive function is comprised of various distinct mental abilities. I'm going to focus on two of them here, which is inhibition and shifting. So inhibition, which is also called inhibitory control, involves the deliberate overriding of dominant responses. And so inhibitory control facilitates self-control, willpower, and self-regulation. Now, shifting, by contrast, also called set shifting or task shifting, involves flexible switching between different tasks or mental sets, and it facilitates efficiently adapting to changing situations. Now, we hypothesize, or me and my collaborators hypothesize, that exposure to chaotic, unpredictable life environments should have specific and opposite effects on inhibition versus task shifting. Now, first off, let's talk about inhibitory control. So, inhibitory control over here is, I conceptualize it as an adaptation to a stable environment. Inhibitory control is about self-control, it's about self-regulation, it's about pursuing long-term goals, and it should be very good if you're in a stable, predictable environment where long-term goals can be realized. By contrast, in a harsh, unstable environment where the contingencies are changing and things are changing, having a lot of inhibitory control might just lead you to miss opportunities that are short-term and fleeting. So a basic prediction from this model is that part of this slow strategy should be high inhibitory control, and part of this fast strategy should be low inhibitory control because they're adapting you to different environmental contexts. Now let's talk about shifting attention over here. Shifting attention, involves flexibly changing. It involves monitoring things that are changing in your environment. Shifting attention should be adaptive when you're in an unpredictable, unstable environment where you have to pay attention to those changes, where the contingencies are changing. And so a prediction here is that individuals growing up under these harsh, unstable conditions should actually be advantaged, should be good at task switching. So they should show superior ability here, whereas individuals in these stable environments where what you want is sustained attention, they will be actually deficient on task switching. So this is specific predictions about the types of cognitive abilities that will actually facilitate success in different environmental contexts. Now, the current research actually investigates the contexts in which older adolescents who experience different levels of childhood stress and unpredictability show enhanced or diminished inhibitory control and shifting. And this is actually, I want to talk just for a moment. My work in this area was actually inspired by research that I was exposed to on rats. And this was research done by Michael Meany, who studied rats growing up in high versus low stress conditions, particularly in terms of levels of licking and grooming and archback nursing they received from their mothers. And what he found was that when he tested these rats from the higher stress parental conditions, low parental investment, when they were tested under standard normal conditions, they looked poor on learning tasks. The rat IQ tests, going through the maze, finding an elevated water platform, they didn't perform very well. But when you actually tested them under stressful conditions, the rats from the high stress conditions actually did better than the rats did from the low stress stable conditions. They actually showed superior learning and memory when they were tested under conditions that were reminiscent of the context in which they grew up. And so this led to, drawing on this research, we hypothesized that childhood adversity is most likely to influence later cognitive function when adolescents are currently under stress. So as a second component of this, we randomly assignment to state stress manipulations of economic decline and chaos. So what we did, in this research, in this research, we had a control and experimental condition. And this was a manipulation of state stress. And in the experimental condition, we had these, these 19-year-olds essentially go through PowerPoint presentations, showing them, they were posed as presenting in a recent New York Times article, showing that the world was going to hell. The economy was going down the tube. It's getting worse. There's no jobs for you in the future. There's a lack of affordable housing. Jobs are going away. We got them. It's called priming for economic decline and uncertainty. And in the control condition, they just looked at PowerPoints, pictures of nature with generic descriptions. Okay. Then in study 1, we had them do an inhibition task. And this is, a very standard, commonly used inhibition task where you, show, here where they fixate on a cross. There's a flash that's meant to distract them on this side of the screen. The flash is followed by the presentation of an arrow. The arrow is then masked, and you're asked what was the direction of the arrow. And you're trying to see if you can inhibit the dominant response of looking at the flash and pay attention to the thing. And so it's essentially the dependent measure is the percent of responses that participants answered correctly out of 90 trials. Now, in study 2, we gave them a shifting task, in this case, task shifting. And this is task shifting where you're shown shapes on the screen, and they vary by color, such as red versus green, and they vary by shape versus triangle versus circle. And in some trials, they're repeat trials. You're classifying a color to classifying a color. Some of them are shifting trials where you're going from a color to a shape. It's harder to do a shifting trial, and so you look at the difference in reaction time between repeat trials and shifting trials, and this is the task switching cost, a measure of task shifting. Well, here is what our actual data looked like. So what do we have? So these initial studies were just done, and these very initial studies were done on college students, not Hopkins kind of students, but from a large public university that have a reasonable diversity of backgrounds, a highly Hispanic serving university, University of Arizona. So we measured, so they retrospectively reported on the levels of stress in their environment. And this childhood stress was, in a sense, this was a continuously measured independent variable. So this was measured, so childhood stress was modeled at 1 standard deviation above the mean or 1 standard deviation below the mean. And then we had random assignment to this control or the economic decline unpredictability condition. And what you see is that under control conditions, when it was just all flowers and sunshine, the kids that had higher or lower levels of childhood stress looked the same. But when they're actually, and this is the inhibitory control test, but when they're actually primed or economic decline, future unpredictability, the kids from low stress environments, they do better. These kids can come from low stress, stable environments. You prime them, the world's going to hell. They bear down and they pay more attention. They have more sustained attention. Their performance increases. The kids from the high risk environments, they go down the tubes, or their inhibitory control gets worse. And this is, this is a significant difference in this, significant interaction here with this. So, consistent with the theory, inhibitory control was something that was accentuated in the low-risk kids under these conditions. Now here is performance on the shifting task. And under the control conditions, the two groups look exactly the same. But when we test them under stress, what we found was that the kids from the higher stress backgrounds, that their performance improved. They became better at task shifting when they were primed for economic chaos and uncertainty. Whereas down here, the kids from the low stress environments, the youth, not really kids, the youth from the low stress environments, they actually got worse. So what you have here, and I just want to say about this task shifting finding, which I think is the key finding, it's also been replicated now in another study using a higher risk sample, although I'm not at liberty to show that replication because I don't own those data. Now, just in conclusion, I believe this is perhaps the first experiment demonstrating the conditions under which exposure to an unpredictable childhood environment leads to improved adult cognitive functioning on a major task, a major, well-established executive function task. And what we found is that exposure to unpredictable childhoods had specific and opposing effects on two major types of executive function. And consistent with the notion that shifting but not inhibition is more useful in an adaptive and unpredictable environments, it was kids from the high stress environments tested under high stress conditions that actually showed this improvement. Now, just kind of thinking about what this means, what the implications of this. To me, these data are indicative that the deficit approach to studying high-risk youth from chaotic, unpredictable environments is not enough. It's not enough, it's only half the story. I partly became interested, I think, in this topic because, as a kid, I went to inner city schools. And even if I saw a lot of my classmates not performing well in the classroom, they performed very well in other contexts. And the idea that these kids are not smart just never had any traction for me. And I think that we need to move beyond thinking about and studying high-risk youth. We need to move beyond this whole approach of what's wrong with these youth. They're broken, they need to be fixed, towards a strength-based approach focusing on what's right with them. What are their strengths? And we need to work with these strengths to design better educational and job interventions. It's awfully hard to change people, to make them different, but it's a lot easier to work with what they have. Now, in terms of this is just, this is a new research program for me, and it has several directions, and one of the reasons that I'm actually took this interview and I'm here particularly interested in a school of education is because there is so much great expertise here in a school of education in terms of doing these kinds of school interventions. And so here are some of the directions that we're trying to go right now. One is just to better understand the functional utility of shifting attention in relation to learning outcomes. So I am working with a learning expert at the University of Arizona to try to understand what good is shifting attention. How does shifting attention, how can it enhance learning? We're looking at, our hypothesis is that it may not be very good for rule-governed learning, but it may be quite good for contingency-based trial and error learning where you have to be assessing changes and contingencies in your environment and responding to them. And so the first thing, if we can once we establish the role of shifting attention in learning, then the next step is very straightforward towards thinking about educational programs that can take advantage of this, where you're using what high-risk youth are good at. Secondly, we want to expand the scope of these state stress chaos manipulations to kind of further understand the conditions. So we're working right now on a classroom chaos manipulation. You got to realize that kids from high-risk environments, they don't their lives are not serene. They don't sleep in serene places. They don't eat in serene places. They don't live in serene places. Why would they be good in learning under serene conditions? Those are the conditions that disadvantage them. So we're looking at things like classroom chaos and noise and conflict, and we're just, again, looking at those conditions. We're also testing this out using a Trier social stress test because, again, kids from high-risk environments, they often have their stress systems activated, and we want to look at learning under those conditions. And then finally, we're also accessing a truly high-risk sample. And we're moving beyond the college students, accessing a truly high-risk sample to enable us to capture a broad range of variation in developmental exposures and to powerfully examine both past and present stressful conditions. So my community partner is a group, it's called Tucson Youth Development. It's an employment agency in Tucson. They are, by the way, very enthusiastic partners. When you actually go into the community and you say you want to study what's right with these youth, you get a lot of positive receptions. People are really sick of pointing the finger and having researchers tell them what's wrong. So this is the group, so this is the control sample are the college students. I refer to them as the control sample, they're relatively low risk. This is the Tucson Youth Development sample. Tucson Youth Development is an employment development center. Obviously you have the same type of things here in Baltimore. It's catering to kids that have dropped out of high school, it's GED training. It is basic job training. So you can see that it's much more of an ethnic minority sample, very different in whether or not they grew up in a two-parent household, much more likely to be exposed to multiple cohabitating father figures, much more likely to have come from, a working class, semi-skilled, 60% versus 8%. You could see the huge differences. 40% of them, their mother did not graduate from high school. All the college students did. And so we have, at this point, we have gained access to this sample and we are working. So one of the things we want to do, because they're seeking job employment, we wanted to do ecologically relevant stressors. So we're working, on a Trier where they do mock job interviews. Job interviews are really stressful for these kids. Mock job interviews, it activates their cortisol, but it's good for them. This is the kind of thing, but this is a real, and we want to look at learning performance under, real live stressful conditions. So just to kind of to sum up my talk, this you can think about as the traditional model. This over here is the traditional kind of cumulative stress, toxic stress, allostatic load, where more danger and unpredictability, more harsh rejecting family environments, elevate physiological mediators, inflammatory tone, HPA responses to stress. This is all the bad stuff going together, like domino pieces falling towards vulnerability to stress-induced illness and low performance in standard tests of intelligence and memory and early psychopathology. This is the whole catastrophe. And this is how people traditionally think about development and stress. And this is the piece, and that's half the story, okay? This is half the story. But this is the other half of the story. And this is the adaptations to stress. And this is when, this is children and youth growing up in these high stress environments. Before these things are going, before you have maladaptation and disorder, you often have adaptations to stress. And this involves cognitive adaptations such as tailoring of emotion systems, arousal responses, perceptual abilities to detection and monitoring of danger. This is increases in shifting attention. This is, insecure attachment. Do you want to promote secure attachment in this environment? I don't think so. This is, more risky and aggressive behavior. This is more a focus on getting it here and now, immediate gratification, early puberty, early sexual debut, lower parental investment, your own offspring. These things are not impairments. These are not dysfunctions. These are reliable, predictable responses to high stress environments. And there's two reasons why this is important. One is that if you want to understand dysfunction, you have to understand function. understanding the functional adaptive responses will help you get a much better handle on the trade-offs down the road, the health trade-offs down the road that result from these, that are often a consequences of these adaptive responses, the trade-offs. But secondly, and equally important, it's important to study these adaptive responses because we can work with those adaptive responses to enhance learning outcomes and enhance developmental outcomes in kids, such as forming learning strategies and educational intervention strategies that actually work with what kids learn in high-risk environments to help them with their learning. Okay, so just to sum up, let me just say a couple words about how I see this Bloomberg professorship. I'm just going to talk generally. I see this, I decided to apply for this position because I see it as an exciting opportunity. I have always been a true interdisciplinarian. I've always been someone that read deeply and delved deeply into many different literatures. I've never had an easy home in any one field or any one department. I'm always an odd duck in whatever department I'm in because no department fits the wildly disparate interests that I have that I try to pull together in my research. So I see this as an opportunity to truly attack big questions about stress, development, learning, and health from a multidisciplinary perspective. And I'm excited about a marriage between public health and education because to me, that is a dream marriage and it really represents the two big things that I'm really interested in. And I have devoted my career to interdisciplinary science. And that's a tough thing to do. When you try to understand something like puberty or understand biological reactivity to stress, and you start reading the really diverse scientific literature, what you find are silos. What you find are silos, people don't know each other's work, there's no connections between them. An analogy I use sometimes is that it's like a library that's been hit by a tsunami, where the books are there, but they're everywhere. And so a lot of what I've tried to do is I've tried to make sense of this. I've tried to reshelve the books as much as I can. But I'm interested in doing more than that. I'm interested in not just reshelving the books, but I'm also interested in leading interdisciplinary groups and really bringing an interdisciplinary focus to tackle these questions, to get out of these silos and really bring together the skills and the interest and expertise that we have here. And I have, for example, I've led interdisciplinary investigators groups on evolution, development, and health. I've led one at the University of Arizona for 10 years. I've really led groups like this my whole career. I have much experience in collaborative leadership, in development of systematic collaborative research capacity, and in attacking big problems from multiple angles. So I am attracted to this because I would love to be somewhere where this is my core job. Not something I have to fight to do, but just something that I'm actually here to do. Thank you. I'd be happy if anyone would like to. Yes, please.
Lovely presentation, and I really like the notion of thinking about one size doesn't fit all,
Yeah.
Particularly when it comes to education. So one of the questions I had, though, and I was reflecting on it when I was listening to your presentation, was you didn't talk about gender. You didn't talk about differences between boys and girls, and adaptation to the environment, and sort of particularly risky environments. And I wondered if you have done any work that looked at differences between boys and girls, or looked at differences between males and females.
Yes, thank you. I wish I could give 10 talks because I have all of my many topics, but that's one of my that is one of my interests and topics. And so in this work, you'll notice that I presented in the developmental model of pubertal development, you notice that it was pubertal development in girls. And one of the things that we find, and has now been replicated many times, is that girls' sexual development, girls' tempo of pubertal development is more sensitive to family and ecological factors than boys. And when we actually study boys, and we have papers where we've compared the two and measured these two things, boys seem to be especially sensitive to their social status and relative position in hierarchies. So where boys are in the peer relationships, boys that are higher in peer hierarchies, that tends to predict for boys. going into puberty earlier, rather, as opposed to these ecological stress factors, which are much more predictive in girls. And so we've interpreted that in terms of, what is important in the environment, that girls' development is very much, in terms of timing of sexual development, is very much more tied to and sensitive to social support, social, ecological, psychosocial factors, whereas boys are tuning in more strongly to their relative position in status hierarchies. Yes?
I'm trying to sort of pull together the beginning part of your talk, and I know that you've only been able to show us the tip of the iceberg of your work. But in the first part of the talk, the dandelion versus orchid, we really come away with the idea that what's adaptive for one group may not be adaptive for another.
Yes.
Or their ability to adapt to their ecology, if you will,
Mm-hmm.
is variable depending on who they are. And in the second part of what you presented,
Mm-hmm.
There's a beautiful sort of acceptance of what the high-risk environment looks like,
Mm-hmm.
and that there is adaptation to it.
Yes.
As a pediatrician in a very high-risk environment, my personal experience is that adaptation varies from individual
Yes.
child to individual child.
Yes.
And many of them may be orchids, and so not really adapting. Even though they are adapting, it is at great cost. And so I wonder
Yeah.
how you're going to bring your orchid versus
Right.
dandelion into the current work, which is trying
Right.
to really emphasize the positive.
Well, okay, great question, great question. If we, go back to that, if you think about that big picture I put up of the two developmental pathways, the fast versus slow, kind of what our work in that area is showing is that being an orchid, being, more biologically sensitive, is accentuating both of those pathways. Orchids are going more strongly down the fast pathway. They're going more strongly down the slow pathway. So they're just more sensitive to their environment, and the effects are occurring for them more strongly. Now, in this second body of research is, the orchid dandelion I've made a career. I mean, I've been doing that for years and years, and have, tried to, look at many, many different variations and alternative explanations. The second part of the talk was was about cognitive adaptations to harsh environments. And in that, that research is in the early phases. To me, it's exciting because there's much, much to do. And in that research, school interventions are all about trying to fix broken kids. And I don't know of anyone who's trying to understand, adaptations to harsh environments and use that. So what we found in that initial research was a main effect. Just as we found main effects in the developmental research before we tested for biological sensitivity to context. Biological sensitivity to context made that research better. Now, I fully presume that as we move down the road and bring in, have a chance to have these kids do TRIRs and to collect autonomic and HPA data, that we'll be able to do a better job of predicting, who is adapting and how. That's, if I'm at step 2 right now, that's probably step 5. Yes.
Talk about interdisciplinary. I think this is the first time I've been in this department at all from epidemiology, but I found your attempt to be interdisciplinary very exciting.
Thank you.
But it occurred to me that your model, there is a contrary hypothesis to your model that you might want to disprove or show where it's right or wrong. So your model, as I understand it, is that there are people who adapt, the people who adapt well to a favorable environment, if I can call it that. I know you don't want to talk about those terms, or
Yes.
environment A
Yeah.
also adapt well to environment B.
They're more responsive to both. They're more open or susceptible to
So
both
the
types of
curves
environment.
cross.
Yes.
But it seems to me that there would be other factors in people that would make them able to adapt to one environment but not to the other.
Mm-hmm. Yes. Yes.
What's
So.
the difference between factors that you
Right. So, we have a language for people that are susceptible to harsh environments. and refer to them as being vulnerable. We have the word vulnerability. Now, in contrast, susceptibility to positive warm environments is called what? No, resilience. Resilience is the ability to experience a negative environment and to be resilient to it. What is it? Thriving. Well, thriving is thriving is close, but is thriving the opposite of vulnerability? So so in 2011, my colleagues, Jay Belsky and Michael Plewis had to invent a term for that other side of the equation. They called it vantage sensitivity. So vantage sensitivity is sensitivity to advantages, whereas vulnerability is sensitivity to disadvantages. So now in this whole differential susceptibility literature, people, there's different ways that these curves can look. And that's actually an empirical question. When they cross over like this, we call it differential susceptibility. They're showing for better and for worse. But sometimes your data don't look that way. Sometimes the data look like vulnerability, or they look like this, and sometimes they look like vantage sensitivity, when they look like that. And there's going to, and that's going to depend on the process that you're measuring and the outcome, and that is the great frontier in this research.
If I may just add one more thing, it seems to me that in the cognitive realm, it may be different from the pubertal realm.
Oh,
entirely, where I think there's a lot of people who can do well intellectually given the advantages, who may not have advantages in harsh environments.
So a vantage sensitivity without vulnerability. We will.
Because cognitive abilities tend to relate positively to each other, I think.
Well, that's the traditional view, and see, this is and when you see, we've used a very middle-class model. We've taken kids, we take kids from harsh, impoverished environments, and we give them tests that we've invented that have been cued on middle-class kids where they take them under conditions where middle-class kids are used to, stable, quiet conditions. Now, I've found, we've been able to break apart some of those executive functions. We're breaking apart shifting and inhibitory control, and we're showing, no, they don't hang together when you think about ecologically relevant contexts and test them under stress. So I question that generalization. Yeah.
I guess I would like to hear you comment on some of the assumptions of the evolutionary perspective, right? So maybe it is adaptive for kids in high-risk, unstable environments to put a lot of their resources into early development, early puberty, and so on, early parenting, but it has a cost for their health.
Yes.
And so
Yes.
from a public health standpoint, if you just follow that through,
Right.
it just gets a little perplexing.
Right. Well, adaptive does not mean desirable. Okay. So we're not. So these are different. So there are responses that I would call adaptive in the sense that kids under stressful, unpredictable environments reliably show these responses. Yes, they're developing earlier. They're focusing on shifting rather than sustaining attention. They're focusing on immediate rewards. From a public health perspective, they are not desirable. But from a public health perspective, we want to address those problems. And my argument is if we want to do a better job of addressing those, we need to understand those adaptations to stress. That will give us the leverage to actually do a better job addressing them. Yeah. Yeah.
We have time for one.
Okay. Go ahead. Sorry. On what?
On blunting, so there's you talked about people who have kids who have a high response versus a low response,
Mm-hmm.
and how there's heterogeneity between if you have high versus low. And I also wondered if you had looked at heterogeneity within those who had low. So if maybe if some of them had low, I guess I was thinking about over the life course people can be reactive.
Mm-hmm.
Eventually, that reactivity dies out.
Yes.
And then have much worse outcomes.
All right.
So I didn't know if some of those kids, if you had looked at who had low, if it was because they were reactive and it was just to their body kind of
Right,
down.
right. Thank you for asking that question. You'll see I'll give you $20 afterwards for asking that perfect question or something I wanted to address but wasn't going to. No. So we have, and for this is a 50-minute talk, but the biological sensitivity context was the original theory that we developed. And in 2011, we published a revision of it that's called the adaptive calibration model. And it is a model about development of stress reactivity. And actually in that model, what we argue is that there are two different kinds of orchids and two different kinds of dandelions that have different developmental history and different health links. This is, my actual, the R01 I'm trying to get them under revision right now is to actually test that developmental model. I mean, that is the way that we are systematically linking susceptibility, health susceptibility, environmental susceptibility to specific health outcomes. So yes, we think there are different kinds of reactive kids, different kinds of low reactive kids, and that it does change developmentally. For instance, we distinguish between what we call vigilant and unemotional phenotypes or patterns, where vigilant are kids that are a kind of orchid kid in a high-risk environment that shows a highly reactive and are very much engaging and defensive around their environment. But over time, that will often shift to hypo-responsivity, where they become unreactive, and it goes from being a defensive to being more of an offensive phenotype, where they're becoming more predators in their environment, and they're not very responsive or afraid of threats. They are the threat. And so that's also hyper and hypo-responsivity are really, in high stress conditions, are facilitating different types of strategies. Thank you.